Hi, I'm Professor Brinton, and welcome to episode 16 of the Notecard Review. Today's topic is a big one. It's that whole pile of enzymes involved with DNA replication. Okay? I have a separate video specifically on the Okazaki fragments and on comparing the different DNA polymerases, but this is intended to be kind of a general comprehensive video of all of the main enzymes. Okay? As a reminder, this process is occurring in a specific direction. DNA and RNA are always read 3' to 5', and written or created 5' to 3', so that we have those ends of the molecules highlighted here. Okay, so this is our 3' to 5' direction on this strand of the polymer. Okay. Throughout this illustration, the DNA is going to be shown in blue and RNA are shown in green. The first set of molecules that come in to begin this process are helicase, topoisomerase, and the single-strand binding proteins. These three are going to work in concert to first open the DNA strand. So the helicase is going to break the hydrogen bonds that are running through here in the middle, joining the two nucleotides together at the nucleotide bases. Okay. The topoisomerase is going to work in front of the helicase to relieve the stress of that coil. So it's as though you had two things here tightly wound together, and if the helicase tries to unwind them without any help, it could potentially break the molecule. So the topoisomerase releases that stress or tension or twisting in front of the helicase molecule. The SSBPs, or single-strand binding proteins, are simply there to stabilize the open molecule, because otherwise you have a whole bunch of nucleotide bases that are exposed here with those partial charges on the hydrogens, etc., that would normally be trying to rebind. So the SSBPs just keep this open for the other molecules to work. The second part of the process is about setting up the situation to where we can add DNA. So that's accomplished by adding a primer. So primase is the enzyme that comes in and adds an RNA primer, shown here in green, and that's binding those nucleotides with the existing strands of the DNA. Okay, so this primase is going to act on both strands, starting at the origin point. Okay, so somewhere off here, off screen to the right on this leading strand at the top is a primer that has been added by a primase as well. Okay. The next step in the process is adding the DNA, which is going to be accomplished by adding the DNA nucleotides onto the 3' end of the primer, okay, on both strands. So the DNA polymerase 3 is the enzyme that's going to come in and attach nucleotides, DNA nucleotides specifically, to the 3' end, bond them there, creating that phosphodiester bond, and start extending that strand over and over and over again, repeatedly adding DNA nucleotides as it goes. Okay. Now on the leading strand, which is shown here on the top, this process can just continue indefinitely until that section of DNA has been fully replicated. But on the lagging strand, shown here on the bottom, this DNA polymerase 3 is going to eventually run into another primer farther upstream back toward the origin point. So that's what's shown here on the bottom with this DNA polymerase here running into this green primer. It can't create this version of the bond, bonding that 3' end of the DNA nucleotide with the 5' end of the primer is just something that it can't do. It has to be able to write 5 to 3 and add the bond that other direction. That brings in our next set of molecules. Okay? In order to deal with this problem of the lagging strand, where these different fragments are created as the process occurs by adding these sections of DNA over and over again onto the primers, the issue of that last disconnect here has to be dealt with, and the primer that's at the origin point on the leading strand also has to be dealt with. Okay, so there's a pair of molecules that deal with this specific portion of the process. Okay, they are ligase and DNA polymerase 1. Okay, so DNA polymerase 1 is going to go through and bind to all of the RNA primers and replace them with the appropriate DNA nucleotides instead, so that that section adequately matches the original strand as well, the original strand of DNA. So this section here that's green highlighted in blue was originally an RNA primer that is being replaced with DNA by DNA polymerase 1. Poly 1 still cannot create the other version of the bond. It can only write 5 to 3. So ligase comes in and accomplishes that specific task. To ligate something is to join it or link it together. So ligase is going to link that replaced primer with the DNA section that has been added by DNA polymerase 3 in the previous step. So it joins the product of DNA pol 3 and DNA pol 1. Overall, that entire combination of processes is what creates the two different sections of newly replicated DNA, matching both the lagging and leading strands that were in the original parental strand of DNA. So this also highlights the fact that we have one new and one original strand in each of the products. So we call that being a semi-conservative process because part of the original is kept and part of it is new. So that's another topic that links into this process. Now, this is very much a flat version, 2 dimensionals. Okay, so there is also a video posted that shows this whole process in 3 dimensions, and I strongly advise that you take a look at that to see the relationship between all of these molecules and what it's doing overall.